hydrophobic. So lipid is actually anything that is hydrophobic. So the hydrophobic things include, we have the triacylglycerols, right? And we have phospholipids, we have the cholesterols, cholesterol esters, and then there's also the vitamins. So you have D, E, A, K. You don't need to know that much about vitamins yet, but just be aware, if there's any kind of disorder affecting lipid absorption, lipid delivery, that's going to be affecting the vitamins as well. Okay. So with this, we're going to start with these compartments. So these are going to be consumed from the diet, and what's going to happen is, let's draw like this. So this is the GI lumen, and the first place it's going to go into is in the, I need to make a bigger lumen here. So this is the GI lumen, and then this is going to be the GI epithelium. And we're going to talk about what's going to occur for these components to get from here to here, and then on the other side it's going to go into the body, so it's going to be taken up out of the epithelial cells and either into the lacteals or into the blood. Okay, so these are the steps we're going to talk about with regards to the lipid digestion. Okay, so first of all, what's going to happen is that if it's lipid and it's hydrophobic, it's not very soluble in water, right? So what's going to happen first is you need something to emulsify it. So I'm going to zone in a little bit. Okay, so the first thing is you're going to need bile. And the bile salts is going to be, which ones are the bile salts that we've talked about? Glycocolate, enterocholic acid, cholate, right? So bile is first. What bile is going to do is going to form into like this micelle. And it's going to be the outside of the micelle. And then, in the environment, it's going to be water, aqueous environment, basically. And then inside the micelle is going to be things that are hydrophobic. So these are going to be hydrophobic things. Hydrophobic things. Okay? So basically, all these stuff that we talked about up here are going to be inside the micelle initially. And then the second thing that's going to happen, you're going to have to break down these components just enough so that they can pass through the GI epithelium. So you're going to at least need some pancreatic lipases. And the lipases don't work completely by themselves, so you will also need the colipase to activate it, and that's going to be located, on the surface here. So lipase, and then maybe colipase somewhere on the surface here. Okay, so now this is the general gist of it. So after they digest the components in the micelle a little bit more, they're going to be able to be a little bit more soluble, so they're going to form into like smaller micelles. So they're going to be like smaller micelles here, and then that would then be able to get absorbed into the GI epithelial cells. Okay, So we're going to talk about the reactions that occur here. We're going to talk about these reactions. So let's talk about the first component that we have on the list, which is the triacylglycerides. And then the triacylglycerides are going to be broken down just slightly. So basically, it looks something like this, and then this is a chain, right? So another chain, another chain. What the pancreatic lipase is going to do is it's going to cut off here, and it's going to cut off somewhere here. So basically, you're going to end up with two fatty acids, right? So that's the chain here. And then you're going to, and you're going to have a 2 monoglyceride. So the monoglyceride, it doesn't mean there's two of them, there's just one of them, but it's a glycerol with the middle carbon still bound to a chain, right? So it looks something like this. This is OH, this is bound to something. So these things are going to be part of some micelles, and then they can pass right through. Okay, so this is in terms of triacylglycerides. Phospholipids, it's the same idea, but it's going to be slightly different enzyme. The phospholipids, so this one is lipase. This is going to be phospholipase A2, and just to refresh your memory of where that actually cleaves, so recall the structure of a phospholipid, right? You have something like this chain. Can everyone see these? Yep. Is it big enough? Okay. And then you have a phosphate group, and then that's linked to a head group. Phospholipase A2 cleaves somewhere right here. And that gives you a lysolecithin. So I'm not going to redraw this, but this molecule is called lysolecithin. And then it needs to get cleaved. It needs to get cleaved one more time to get rid of the other chain, and then that's called the lysophospholipase. I thought it was I'm pretty sure it was the phospholipase A first, and then the lysophospholipase, because this one is technically considered lysophospholipase, so it would make sense that the lysophospholipase would come and cleave that one. Does that make any sense? Yeah. Okay, so after all that is done, you basically end up with a molecule, phosphate head group, like this, and that's a glycerol phosphate with a head group. So I'll abbreviate glycerol phosphate. Glycerol phosphate. And then you're gonna have the two fatty acids, and then all of them are gonna go into the GI. Okay, so that's the story with the phospholipids. And cholesterol is, I guess, a little bit more straightforward. You just have so cholesterol that are in the form of cholesterol ester is going to be turned into cholesterol. So just to remind you the structure of cholesterol, you have something like this. You have a ring. You have several rings. Oh, sorry. Okay, so basic structure of cholesterol, but over here for cholesterol ester, it's attached to this group. So this is cholesterol ester, and this is not as soluble as if you have the whole molecule attached to a OH group. So that's I'm not going to draw the whole thing again, but this is the whole molecule, but basically the part that's in red is now attached to an OH group, and that's the cholesterol. And this is the cholesterol esterase. Cholesterol. Okay. So now all these, this is cholesterol. Okay, all these molecules get taken into the epithelial cells. All these reactions here that we talked about is just so that they're just soluble enough to be uptaken into the cell. And then once it's inside the cell, basically the reverse is going to happen. So this is going to form back into a triacylglycerol. This is going to be back into a phospholipid, and then this is going to be made into cholesterol ester again. Yes?
For the triglycerol, so in literature, for three fatty acid and glycerol, is that the same thing like 2 fatty acid and
Yeah, so basically what happens in the GI lumen, they don't cleave off all 3. They only cleave off 2 of them, and then they leave the other one on, and then that gets transported into the epithelial cell and then forms back into a triglyceride. Not quite sure why that's the case, but it only cleaves off 2 of the chains, not all 3.
Because I think in physio last semester was all 3 or something like that, right?
I think it's 2 chains. They weren't very specific in physio class about exactly which ones were cleaved. So, are there any questions?
That's it.
Okay. All right. So you can basically summarize all these reactions as to basically increase the solubility, just slightly for uptake, right? And now when it's inside the GI epithelial cell, all of these are going to be put back to what they were before. But in addition to that, we're also going to make the chylomicrons. And what you need for the chylomicrons is this ApoB48. So I think of it as basically the structure that kind of helps to organize everything into a chylomicron. So the end result is you want this to come out as a chylomicron. And then the chylomicron can go and drop off things and then go to the liver. So we'll talk about that a little bit more later. So in your epithelial cell, you want to make the ApoB48. And where the ApoB48 comes from is the I think it's apo gene. And this one gets made into ApoB48. I'm mentioning this here right now is because all the ApoBs are made from this same gene. So later on, we're going to talk about the VLDLs, IDLs, and LDLs. So that also contains an ApoB, but it's a different form. So this one is capable of making it into the ApoB48 and then ApoB100. And then, is there any question? Yes?
Is there no gene for the ApoA?
It's a separate gene. Yeah. It's an entirely different gene. So ApoB is only going to make the 2 forms of ApoB proteins. So you can imagine if there's a gene defect for ApoB, let's say, that's going to affect things that only have ApoB, right? So chylomicrons, VLDLs, things like that. But we'll get to later, but HDLs, which doesn't have this, they only have ApoA, is not going to be affected by a gene defect in this. So just be aware that it came from the same gene, and it can have two different products, and I believe it's the RNA editing. So one of the nucleotides gets changed, and then it makes this product. Okay. So now to assemble the chylomicron, you have this ApoB48 made in the rough ER. And then all these are kind of getting taken up and then repackaged in the smooth ER. And so all these products here will have to get transported into, along with the ApoB, into a chylomicron. So they will need to go, these products will need to be transported in, and this is mediated by the microsomal transfer protein. So this is pretty much essential in making the chylomicrons. Otherwise, you'd just be making, those apoprotein, and then nothing is coming in. So basically, you won't have a chylomicron being made. Yes, other question?
Yeah. It says there's microsomal triglyceride transfer. Does that mean this still takes up all of them?
I think it's responsible for all of them. I think one of the diagrams just shows like different components being transferred in with that. Yeah. So I think the name just says triglyceride, but I think it's all of these components require this protein to be transferred into the chylomicron. Okay. So now with ApoB48 and all these components, it becomes a chylomicron, right? And then this one is considered a nascent chylomicron because it doesn't have other things yet. But the job of the chylomicron now is to basically drop off a bunch of triglycerides and then whatever's remaining, so mostly cholesterol and cholesterol ester, that's going to go back into the liver. And then we'll talk about it a little bit in the next one. So what I want you to take away from this whole thing is what's occurring in the GI lumen. So basically all these can be summarized in this. So it can be summarized by what you're doing there is increasing the solubility just enough to uptake all these lipid-soluble food material molecules. So with the triglycerides, it turns into a 2 fatty acid chain, M-monoglycerol. For phospholipid, it turns into 2 fatty acid chains and a glycerol phosphate. And cholesterol esters gets made into cholesterol, and then that's taken into the epithelial cell and converted back to what they were, and then everything is packaged into a chylomicron along with the ApoB48. And then the chylomicron will leave the epithelial cell and then drop off the triglycerides. And then the rest of it will go back to liver. So this is a very general, broad picture of what happens with lipid digestion. Are there any questions? No? Okay, I'm gonna pause this. I'm gonna save this video first.